The agenda being rather packed, I'm going to try to be as quick and efficient as possible. So this is feedback on a workshop that was held at INRAE Versailles near Paris on a large language model, on knowledge graphs, and especially on two technologies, BioChatter and BioCipher, that are general bio-adapted tools, but we wanted to explore them for plant use cases. I organized this with Rafael Flores from URGI, Sebastian Bayer from Jülich in Germany, Pedro Barros in Portugal, and Sebastian Lobentanzer, who is the BioChatter and BioCipher expert on PI. He's working at the Institute for Computational Biomedicine, Heidelberg University in Germany, as well as at EBI and especially within the Open Targets group. There were three objectives for this workshop. First, to build common workflows using BioCipher, AI knowledge graph framework, and the breeding API. And they extend this to other sources, CorCocDB, Narrow in Japan, or other sources, as you will see. And on top of those knowledge graph, deployed BioChatter for building something that is more biologist friendly, or at least more general bioinformatician friendly than a raw BioCipher interface. And finally, the last objective was capacity building for all attendees on knowledge graphs, on artificial intelligence, LLM, on BioChatter and BioCipher. So what's BioCipher? So it's mainly, in my opinion, it's a workflow. It's an ETL tool written in Python that brings reproducible Neo4j knowledge graph builder. It produces simply this from any input, as you can see. Sorry. You can use CSV file, you can connect to HTTP. In fact, whatever source that is connectable using Python. And this is why the language is important. There are also some ontologies connections which are important and not necessarily that important, as you will see. BioChatter is the second part. It can be plugged on the BioCipher built Neo4j graph. So the important thing is that plugging BioChatter on any BioCipher, on any Cipher Neo4j graph is difficult, is tricky. This is really one of the results we got from this workshop. BioChatter can work with any GPT engine, with Olama, with any open source one, with the Mr. AI one, and so on. But of course, it is highly optimized with OpenAI, with ChatGPT. But it works best with this OpenAI API. It provides both a web interface and the engine to do the actual querying, or in fact, the multiple possible queries. So there are several workshop use cases. The first one, on wrapping, we worked with two data sets extracted from the breeding API endpoint from URGI. As a reminder, breeding API is a web service API developed for 8 years, something like that, with the CGIAR, Elixir, INRAE, well, a lot of people. And it provides a way to access germplasm data, so genetic resources, phenotyping data, and genotyping data. So our two data sets were on Poplar on the WheatIS serial network. We had several questions in mind, scientific questions. For instance, what are the trees older? Some 12 years in CSV format with location, etc. Which variety showed the highest shoot dry weight in Clermont-Ferrand? What data sets allows to study relation between flowering and yield or disease? So all of those questions are really geared toward extracting data set to prepare further analysis. So we tried to generate, in fact, we succeeded in generate knowledge graph using BioCipher, and plugged the biochatter on top of it. It was quite easy to set it up and to configure it. It took me a couple of days, and I was quite rusty in my Python, so that's encouraging. And there is also loose coupling to ontology. And in fact, there are two stages of ontologies. Foundation one, biolinks, that provides a core concept, as well as one dedicated to the data that you are trying to import, which is much more loose, which provide an adapter-specific graph data model, which is super convenient because it allows you to pick only the concepts that are relevant for you. Then we use BioChatter to generate breeding API queries. Instead of having a knowledge graph, we have the possibility using the chatbot to get a series of BRAPI query to get the data we want, like give me all the Apache or Bonfermier variety studies. And the chatbot was able to generate very convincing BRAPI queries for that. So the big advantage is that it uses the API usage, so BRAPI usage, which is not always super quite forward, so it's a good thing. But the weak point is that the chatbot was easily lost when there were needs to chain multiple API calls. So for simple use case, it works. For more complex use case, it's less convincing. We also try to change the BioLake Foundation ontology of BioCipher with the MyAPI ontology, so PPO. It was really not straightforward. It generated a lot of side effects. We didn't really succeed during the workshop. And after a couple of days of trying, especially with the Portuguese team, I've got the feeling that this is not necessarily a necessity. It's a lot of work with little gain from the BioCipher, BioChatter building point of view. To be able to correctly tune the chatbot effect, how it works, the adapter-specific graph data model is much more convenient. So a few screenshots of what this BioCipher, BioChatter can do. So here is the very rough chatbot interface that gets generated. So with a simple Docker Compose up, so it is fully Dockerized. So once configured, we can set it up in less than a minute. Here you see the answer your question, so the prompt entry, the schema info if you want more information of how the data is modeled. And here an example of the answer to the response. What are the studies that are using Apache? Apache being a variety. So the chatbot generates a Cypher query here, run it, and get the result as JSON that can be used then for anything. So there have been many other use cases. I'm not going to go in a lot of details in all of them. First, because of time. Second, because I'm not super comfortable with the others. But if the camera is not wrong with me, I think you will be able to discuss it among yourself. So there has been a plant translational database with autologous, with WheatIS and Arabidopsis, with gene homologies, ontologies, transcript. They succeeded in building the BioCipher instance plus BioChatter to generate Cypher queries. There was also a group working on mobile element knowledge graph, as well as stress knowledge map use cases. and the chem and plant knowledge graph use cases. In all of those cases, they wanted to reuse existing knowledge graph in Neo4j. It was not super conclusive, not very easy. If I understood things well, the stress knowledge map succeeded in plugging biochatter on it, but that was the only scenario. On the other scenarios, the conclusion was that it's better to build a knowledge graph from scratch using BioCipher rather than plugging biochatter on an existing one. Finally, there was the tomato knowledge graph use case, which was the extension of an existing BioCipher knowledge graph to add biochatter on top of it. So that was rather straightforward. He experienced several fine-tunings. For instance, in the model, he showed that using age labels such as regulates instead of transcription factor regulation, the later one being more ontology compliant and the first one being simpler. In fact, the first one regulates it better because this is the one that is used by biochatter to really identify the age that it has to use in, in the Cypher queries. So to try to summarize this a little bit, so regarding BioCipher, my personal opinion is that it's robust. It uses Python, rather well-written one. It has poetry dependency management. It is highly Dockerized, very easy to run and set up. It proved to be really a very reproducible extract, transform, load workflow. We have to write ourselves, sorry, our data adapters. And that's a potential bottleneck. First, of course, because we need time to do this. And second, because we need to optimize them, especially for huge data sets. For genomic data or things like that, with a lot of ages, we cannot load all of this in memory before loading into the Neo4j database. So there is a need of a few commands to set up a working instance. So it was really a breeze on my laptop and has to be tested on servers or Kubernetes. And you can see here on the right an example of a BRAPI adapter with the pre-pros data that opens the different data files, and then once it's all loaded in memory, fills the nodes file for loading in Neo4j. Although BioCipher includes the simplified data model, as you can see here in this YAML file, you've got this trial object of type dataset, the dataset being declared in the foundational database with its list of properties, the field that it has to take into account, and there is also a definition of the edges later. Biochatter. My personal outcome is that it's absolutely not magic. That the idea to have one single big chatbot that solves my problem is not going to work. And that, in fact, we've got to aim for multiple chatbots, programmatically chained, as we can see here, with first a question in a natural language that is interpreted by the chatbot that generates another question or a Cypher query, as we have seen, that is then run to produce the results that can be summarized using another agent or the initial chatbot. As we can see here, so this is exactly what I just said. The other thing is that if we really want biochatter to give precise question, we can fine-tune it, we can over-optimize it, but therefore this over-tuning makes it not maintainable on the long run. So this is not a desirable approach. And I'm done with the feedback, and here are all the people who participated in that workshop.
I can start. So just on BioCipher serial, so we are interested in probably looking into a new ETL tool for Netminer as well. So do you think there is, what would you recommend? Is BioCypher good enough for building knowledge graphs, like for example, Netminer has, with variety of data sets, crossing ontologies, hundreds of thousands of publications, ensemble genomes, can scale to that sort of use case as well?
So, Kieran, I know that you are thinking about updating your ETL. I think personally that BioCypher ETL is very convincing, but there is a need of more testing for its capability toward a huge volume, because you have your huge graphs, especially for the data adapter. For my case, on most scenarios in the room, they were quite small. Here we need optimization, such as not loading all the files in memory, but streaming them before writing their output and so on. Nonetheless, I think it could be really worthwhile testing it. And my advice would be really to get in touch with Sebastian Lobentanzer, so the BioCypher PI. He would be super happy to work with you. I'm convinced of that. If you want me to put you in touch, I'll be happy to do so. Or if you've got any connection or already using any other mean, of course, please do so.
Thank you.
Other questions in the audience or online? No? Yes,
I'm not sure to have understood if there is a kind of built-in model in BioChatter, in BioCypher. Does it imply to have his own model?
So there is a foundation ontology, BioLink, that is used at the loading stage. Here it is. For instance, here you see the word dataset or the word study are taken from this foundation ontology. But it acts more as a reference vocabulary, more than as a data model. In other words, you need to have just a couple of roots in this BioLink foundational ontology, and then you elaborate your model directly in your configuration file.
Okay, so the constraint it comes from this ontology, in fact, this foundation ontology.
Exactly.
That means
exactly.
that if you adapt this to an existing knowledge graph, you have to change things. In fact, in this, you have to rewrite maybe the foundation ontology to be able to feed the final graph. That's right.
Yeah, we tried, in fact, simply to replace this BioLink ontology with the MyAPI ontology, which technically is quite feasible. We were helped by Sebastian. It worked at first. It loaded in BioCypher, so in the knowledge graph, but it had undesirable side effect. In particular, it was much more difficult to steer how BioChatter would generate the Cypher queries if we removed the BioLinks ontology. I'm pretty sure it could be doable with a few extra days. We didn't have the time. And it's more, I don't see today what would be the interest of doing so. Because using this configuration file, you set up your own data model.
The problem comes from BioChatter. That's right.
BioCypher and BioChatter, there are a lot of cross dependencies. to have BioChatter and BioCipher work seamlessly together, they have really to be built together and feed together. Hence the interest of this one command that generate both in a single shot.
Another last question.
So you're looking at chaining multiple chatbots, and chatbots have got a tendency to hallucinate and bring errors into them. Are you amplifying the errors by chaining together multiple chatbots?
Excellent point. And this was my biggest worry when I began to work on that. My impression today, and I'm super cautious on that, my impression today is that with little, very narrow chatbot, we in fact minimize a lot the risk of hallucination. So it's better to have a series of little chatbots, very little prone to hallucination, rather than bigger ones that could more easily state wrong things.
I'd be interested to see the results of that. It's an interesting idea. Thanks.
It's first tests, but
Yeah.
Okay, thank you very much, Cyril, for this presentation and staying with us at night in France.
All right. So yeah, since we have a very short time, I'll just give a very kind of a lightning talk about GradGIS, what GradGIS is, the content that we add, and then I talk about the AI the things we are doing. So GradGIS, I think most of you know, but some of you may not, is actually the US state funded project. And we have been around since 1992, so it's definitely over 40 years. So that's your state foundation. And we serve as a centralized place for the international global data management system, and we provide digital platform. I'll go very fast, okay? So just don't, essentially our user base is global. So maybe search and browse. We have an area you can click on it, and there are different ways of searching. But the important thing is for this group, search WheatIS box. So you can put a keyword there, and then you can actually go to WheatIS, and from WheatIS you can come back to GradGIS that way. Can people hear me online? Okay, so if there's a problem, they will let us know. So yeah, some of our accomplishments, creation is a big part of what we are doing. So just like this a yearly basis, we added almost 6,000 QTLs and other type of genetic and genomic content.
Sorry, that's me.
So yeah, so we have actually right now also the tools that we have 210 genome browsers and 408 plus databases. So this is one area that we are strong at. For the content, this is not this is in the PDF format, but in October, Bob McIntosh created this WheatIS catalog, and he covered all the WGC creations up to that point. I don't know how he does it. He's amazing in that respect. So this is a curated G catalog for wheat, and it has been around for decades now. Thanks to Bob McIntosh, it's been unified. So I said we have genome browsers. Recently, we actually made available Barley Pan Genome V2. There are links there to go to Pan Barlex, for example, at IPK. So we also have Civil 2 available under Toronto agreement, and the Panot browsers that we developed, but is not under that. We do have the pan genome areas as well, separate. So this is Einkorn. We have several tools for JBrowse2, BLAST, and other types of tools. We also developed an area for AgileOps Stash, and a separate with genome is being area is being generated. We like to, because we do have lots of genetic data, we like to integrate or link genomic and genetic data sets. So this is one example of where we have the GWAS set aligned to GWAS QTL sets aligned to genome browsers. So that's one track. Whenever we can, we do that. Then you can get information. We also added tracks for WheatIS collection. So this is these are huge sets, so we weren't able to include everything, but at least we created tracks so that we can get some information about the work that was done and generated. We have links. One of our close collaborators is 3TC Toolbox for genotyping and phenotyping data. So we go to T3 from our genome browsers, but T3 is not the only place we go to. We go to XFIT, Netminer, Ensemble Plans, and the commercial genome browser called Persephone. So we recently this year, I think, added several tracks to go to AlphaFold. We cannot compete with Ensemble Plans right away, but we do things in our way, in our focus. So you can get more. And the nomenclature and data standards are very important. I think also it's a focus for this group. So last year, genome nomenclature, new updated guidelines for genome nomenclature is published. I also mentioned the OAT nomenclature paper, and then we have this, as I said earlier, we have this page where people can get codes, identifiers for their species and cultivars. So this is under development, but this may be of interest to Cyril, maybe, if it is still online. I don't know. We are actually developing this search area where people can actually put keywords and then put other graphically enabled resources and get some results back. We do have some databases, some data types right now available, but this is under development again. And we have problems actually reaching URGI for some reason. So we'll contact Michael and see about this. Okay. All right. So going back to our site, so we have the curation. We have also created data. We have working decades on work, and of course, Laurel was one of some of the curation of Laurel that was amazing. We are very lucky to have this high level PhD level curators. But this requires usually PhD level domain experts. And these are very breeds, right? So without curation, I mean, essentially here it's pretty required. We know the importance of curation. Content size will be low, data accuracy will be low, linking will be more sparse between data types. So what to do? So we thought that maybe ChatGPT or other LLMs can be a way to reduce the need to do more curation, also to be able to allow us to do more curation. So we just focused on the QTLs and PAs, and this work is actually essentially done by Eli here in Koreski. So I am essentially presenting his work here. So yeah, we essentially gathered a bunch of PDFs that we know what it contains because our curator went through it and curated the data from there. So we knew what we can use as a validation set. So we applied the ChatGPT's models to be able to extract data and ask ourselves whether it can extract data. And also look at the prompt engineering and the hallucination that they actually brought up, hallucination issue that they brought up. So we looked at the first, the abstracts, whether we can actually classify abstracts, and we use these categories: agronomic, biotic. They're a bit overlapping, of course, but essentially our classifier was able to classify this very nicely. It clustered in different clusters. So that was good. We saw interesting that here GPT 3.5 did a bit better than the GPT-4, which was interesting because GPT-4 is a newer version. We also provided this prompts. You can see the prompts on top. And also this paper will be published soon in database, so you can have access to the whole the graphics, etc. So we essentially said, can we extract genetic information? And here GPT-4 did better than GPT 3.5. So that was good. We also tested whether hallucination may play a role, and we forced GPT to hallucinate by asking peduncle length. So peduncle length, for those like me who do the studies, so peduncle is that area, little area, like there's a panicle, which is different. So we essentially forced GPT to hallucinate it, but GPT-4 was very nicely captured the peduncle. You can see this, not GPT 3.5, but 4, it was good in getting the positive counts. So that was impressive for us. So prompt engineering is critical. I tell people that when Google came out first, if you remember, people were asking, how will I search this? Right? There was a little learning curve. It is the same with this prompt engineering with using the GPTs. You have to know how to ask the question. So here we say identify all QTL, trait QTLs, blah, blah, and then we provide some kind of instructions. So that's the orange part. Okay, and then there is this underlying part. So range, include all the traits with QTL located on such and such area. So that's the instructions and range, right? So we tested this, which one is actually will that make any difference? And yes, it does. If you look at the GPT-4, full and range will be the underlying area, and then orange is with the instructions and blue is without the instructions. And you can see that the number of correct answers are just so different when you ask the right question or if you provide additional information. That is something to think about. We don't usually think about like how we write the keywords or questions. For example, WheatIS, we had keywords, right? But how would you ask the questions? I think we should put something when we develop this, some guidelines to users saying, hey, this is how you can ask questions, so they have an understanding. So take-home messages, GPT-4 is definitely very powerful, and new GPTs will be much powerful, I'm sure. It was able to, GPT-4 was able to retrieve 91% of disease-related human-created QTLs. That's pretty impressive. It's not complete, so you cannot always get the whole picture, but perhaps for your application, 91% will be good enough. So that comes with a grain of salt. So GPT-4 is definitely better than 3.5, consistently, not in every text. So LLMs demonstrate the potential to extract and present information. So it's upcoming. I think that we will use its power more often in the future. So, okay, so thank you. So we appreciate the generators, curators, and our advisory committee, and thank you, our collaborators.
Thank you very much. Very interesting application of AI.
So I wasn't quite sure what to talk about, so I've basically thrown together a few different slides on some of the different areas that we're working on. We are short of time, so I'll go through very quickly, but be happy to talk to people afterwards if I can sort of things that we do. So looking at, we've done a lot of work on machine learning, and as you'll know, machine learning is, you can do things like take image data, text data, tabular data, you put it through models, build models, and you can use it for classification, regression, and clustering analysis. And this is where we'll use most of the AI type work. I've done quite a lot of work on image analysis, and I should apologize now that there's almost no wheat in here at all, because I actually do very little work on wheat. But some of these things can be extrapolated to wheat if you use your imagination. So this is a problem that we had in Australia, where you've got sandplain blue lupins as a weed in narrow leaf lupin crops. There's obviously no chemicals that can separate them. So being able to identify them from camera images to be able to do targeted spraying, we built a model to go ahead and do that, and you'll see it works fairly accurately, even though the crops actually look very, very similar. The model can pick up with high accuracy. We've done quite a lot of work on canola. Canola's one of the big crops that we work with. And here we kind of flipped it around, because rather than trying to say, okay, let's try to identify a weed, we said, well, there's so many different weeds that you get canola. Let's just see if we can just identify canola and assume that anything else that's green is a weed. So this is a model that we built here. And as you can see, it clearly identifies, I think it's mostly grass weeds, but plus some other weeds in there as well. And even in quite complex soil environments with a lot of stubble and things there, different stages of plant growth, it works pretty well. And that was just published late last year by a very talented master's student who's now trying to solve cancer, identifying cancers. This is my wheat slide. We did actually do a bit of work on wheat. This is identifying salt stress using hyperspectral drone data in the field, working with Mohammed Ben Moon, is one of the first collaborators I've been working with for quite a while. And that seems to work pretty well on field data. This highlights the fact that I'm not a machine learning expert, but I do work with machine learning experts such as Mohammed and others who make sure that we're on track, and I keep him on track by saying, actually, no, this is how it works from a biological perspective. So we've got collaboration. We've got quite a few papers out together on this. We also do a lot of genomic work. So this is predicting gene models, trying to identify whether it's an accurate gene model or not, putting in various parameters. I think this is mostly using legume data, but it can be applied to pretty much anything, and it highlights one of the things we've been looking at in terms of when you look at gene annotation, the prediction of gene models. They're really variable depending on who did it and the mechanisms that they chose. So we developed an approach that's published in a different paper to say, okay, based on conservation, these are real genes, these are not real genes. And then we build a machine learning models with a range of features to be able to say, well, these are the sort of features that will predict with accuracy whether it's likely to be a real gene or not. So we can go through and use this approach with the other approach of conserved genes to be able to look at your gene models and be able to say, okay, yes, they all look really good, or maybe you should look at some of these ones again just to see if they're right. This is using relatively basic information going in to build a model, and I think we could do a lot more with this. AlphaFold would be really unique, considering flexibility is one of the major factors here. Trade prediction is obviously very important. We've done a lot of trade prediction from genomic data and from image data. This is an example we published a couple of years ago in soybean using quantitative and qualitative traits, assessing machine learning and deep learning methods. The machine learning outperformed deep learning, but that was presumably, we're presuming it's due to the quantity of data. We had 5.5 million SNPs, up to about 1,000 individuals per trait. There's a lot of data in there, but I think even still, there's not enough for the deep learning models that we're applying there. This is looking at phenotypic assays in canola to try to predict which major resistance genes are in canola. These are normally assessed by doing infections on cotyledons. An interesting thing that came from this is that we had 4 replicates for each of the samples that we put in. And we thought, well, do we group them? Do we average them? Do we put them in separately? Do we put them in separately? The model came back and said, oh, replicates 1 and 2 are really good. Replicate 4 is crap. And that really got us scratching our heads because they're replicates. And it took us a couple of months to actually realize, from talking to the person who did the assays, was that she did the assays, and if 3 of them were the same and maybe 1 of them didn't infect, she put that one in as last. So the first 3 scores are this, and the last replicate is a dodgy one. And the model picked that up. So it picked up the fact that the data wasn't as standard as you would expect. So it's quite nice the way that works. Some of these genes are quite difficult to predict using standard methods, so this is really quite useful. This is an example of taking genomic data and looking at quantitative resistance in canola to blackleg. It's interesting because it's a relatively small data set, but it's a very clean data set, and we get new data for these trials every year, and the accuracy improves each year. And even though we've tried other assays, other analysis with much larger data sets, we often don't get as good a result as with this. Also, as each data set comes in, the data changes slightly, and the type of model that works best tends to vary. So XGBoost tends to work quite well. Random forest sometimes comes out on top, and it really depends on the data set. So you can't really say this is the best approach. You've got to basically just try them all out and see which works, even for data sets that don't change a vast amount. The loci that were important for prediction, they also overlapped with gene loss. So it's quite nice validation. We're doing quite a bit of work on yield prediction in maize. We're doing all these weird species because data is available. If someone's got a whole heap of wheat data, I'd love to be able to play with it, but we haven't really seen that much. This is trying to predict yield from relatively young maize plants, image data, plus some other small amount of genetic data, and we've started building models with environment data as well. The actual yield is quite difficult to predict, but classifying it into high, medium, low comes with quite a lot of accuracy. We were part of an international challenge to be able to competition. We certainly didn't win the competition by far, but the people who did win the competition was interesting because they were not like us. We tended to throw everything into the model and got the model to sort it out. They actually did a lot of manual curation of the data, and especially resolving the environments and normalizing for the environments before we put the data in. They had much higher prediction accuracy. So we learned quite a lot from that collaboration. SNPs. We're moving away from SNPs. We realize that SNPs have got a lot of limitations, especially for predictions. There are far too many of them. It causes problems for machine learning predictions. We're moving towards haplotypes. We've developed this tool that will analyze haplotypes at a locus level. So you identify QTL, you throw the region in with the trait data, and it will start to resolve the haplotypes and tell you which haplotypes, and then it will actually start to identify which particular changes are likely to be causal changes. This works on a locus by locus way. We analysed quite a few examples of this now, either published or in press. But this is a major protein locus that controls the oil protein in soybean with a high level of accuracy, and that was a really quite nice result. With the haplotypes now, we're looking at genome-wide haplotyping. So rather than going through and looking at individual loci, we're trying to be able to say, okay, here's a population, here are all the SNPs. Can we resolve the haplotypes in that population? And instead of putting in SNPs into the trait association model, using whichever method you're looking to use, you're actually putting in haplotypes. And we think that this has got a lot more power than using SNPs in itself. Again, we're not really doing this in wheat yet, but we hopefully will be in the next 6-12 months. Large language models, we've been playing with these for about 18 months, 2 years now. We haven't really had any funding in it, so we've just been basically playing around the edges. We've been looking initially as a test in legumes and looking at disease. So we've got a few thousand publications. We initially just put the abstracts and built a model using the abstracts, and asking questions from that seemed to work pretty well. Gave us a reasonable amount of accuracy, but it certainly didn't capture all the information that was going to be in the papers because there's not that much there. We've found if you just throw all the papers in as they are, it just it gives you garbage back. It's really doesn't like it. So we basically download the papers from the publishers, and we've only done it with a few thousand at the moment, but that could increase quite quickly without too much trouble. And we format them. And what we're seeing is it's really quite important to be able to structure the data here. And this is one of the things that we're working on at the moment, which is why I was asking you about the chunking, is that if we can structure and annotate the manuscript, then it's going to give you far more reasonable answers from that. So we've been working with a researcher in the University of Melbourne. She's moved from University of Western Australia to University of Melbourne, and she's been developing query systems for things like car navigation and stuff like that, and lots of weird data formats and military stuff. And she's found ways to be able to say, okay, you get a PDF of a publication, it's got images, it's got text, or you get a newspaper with images and text, and actually link the images with the text and actually build on that information. So we're working with her to go through and start to annotate the papers. The methods that we're going through is that we annotate a few hundred papers first, in terms of saying, okay, this is this type of sentence, and then use that to build on models that she's got that will go through and then automatically annotate additional papers, so that you then have that resource of annotated papers that could be fed into the model with much greater accuracy than otherwise. So yeah, we've got to instruct it not to guess. Something hallucinating is going to be a bit of a challenge. We've got to be able to do it so that we have new publications as and when, and also to highlight conflicts. So there is a conflict between papers, which happens quite a lot, to actually bring those out. We still don't have funding for this at the moment. It's mostly been run by Monica, who was a PhD student in my group, postdoc for a while, but now she's setting up her own group. So she's leading a lot of this still. So she'd be the person to talk about this. She's got a master's student who's got about another 6 months to finish this project. So once that's finished, if anyone's interested, let me know. I'll pass on the master's thesis to see how long it goes. It looks like we will get some funding in this space. We've been shortlisted for a GRDC funding application AI breeding tools. That's together with University of Queensland and CSIRO. This is more general in terms of breeding tools, and there's not a vast amount of funding, but it gives us a foot in the door, and it links quite nicely with some of the tools that CSIRO have been developing in this space. We think there's potential to start implementing some of this into the information system, developing large language model interface for big publications, and hopefully down the track at some point integrating genomic and trade information. And I didn't do any of this work. I've got a great team of people who did. So I've tried to acknowledge them as I went through. Happy to take questions.
Thank you very much, Dave. So, questions?
Yes, I have one question about the way you use AI to determine the good gene model. What is the parameter you use? You speak about the flexibility of models, but what are the other, the number of exons, something like that, size of exons?
So the most important one is, and it's not from the AlphaFold predictions, it's just some prediction of flexibility just on the gene. This is quite a basic project. Shri did it as part of her masters. She's now doing a PhD degree in this space. So length, molecular weight, which is related to it, they're important, and then various percentages of amino acids. I think combinations of amino acids, I think it's going to be more important, and I think there are probably a few other parameters we could put in. This is a very basic model, and I think there's a lot more that can be done in this space. The important thing was initially identifying what we thought were good genes predictions and which are bad gene predictions, because otherwise you're just reflecting what you're doing in terms of that choice, and that was based purely on conservation.
Okay, I was going to say that was going to be my question. How do you know which are the right genes? That's always.
So if you take 10 legumes, which is what we did, the annotations for those, we looked at genes that were predicted in one legume, had no sequence identity to any part of the other genomes, had nothing in the nucleotide database, we said that's likely to be John.
I will push back on that one.
Yeah.
Right. I mean, there's definitely unique genes for different varieties, and that's where the treasure is for breeders.
Yeah. So you say unique genes for varieties, but if that gene is only in that variety and is not in any other wheat variety and it's got no sequence identity to any other plant gene, there's a good chance that that is not real.
Oh, you may.
So it'll be unique to that variety and that'll be a value, but there's a good chance that that will be in other wheat lines, that will also maybe in oats or something else.
I see. When you say you make it 10% or something, the signal. I see.
Yeah. So it's got to be you, and as you can extend it. We only did it with 10 initially, but you can extend that to a much larger number.
Those kind of novel genes also wouldn't have AlphaFold models because it's kind of based on the conservation, so they would be lacking supporting kind of predictions from that also.
Yeah, but we're relatively easy to make models for those and have a look. So I think that was quite nice. Another master's project. So I run a master bioinformatics course. If anyone's got some data sets that was to analyze it, throw them at it. So
I was just repeating. Yeah.
Just to think, what kind of machine learning techniques is behind this?
I have to go back and look at the paper. This is almost 3 years ago. It's very interesting.
Can you send me the paper?
Yeah, it's published. It's there.
But I know the UI or something. We will have
Hold on, I'll get the full reference.
Yes. Another questions or remark?